Thanks, Hongwei, for the introduction. So when I was preparing for this seminar, I realized that, this is a very special seminar because it's the first time that I ever present to my Tsinghua colleagues and students as a professor from Princeton. And I also am very grateful that Hongwei and his colleagues arranged my talk to April. April is really the celebration month for Tsinghua for the anniversary, right? And also for our department of Biological Sciences and Biotechnology. Now it is the School of Life Sciences. And also for the title, just to echo what Hongwei said, there and back again. So I have been following a ping pong trajectory, Tsinghua, Princeton, although I don't know where my next stop would be. For sure, there will be another stop, maybe, I don't know in how many years. All right, so as Hongwei just mentioned, since 2007, my lab has been focusing on the structure mechanistic investigation of membrane transport proteins. So because I have prepared too many slides, I realized, so I will skip this introduction, but just simply put, membrane transport is the essential physiological process. And by selecting, the physiologically and pathophysiologically important transport proteins, including both channels and transporters, we try to reveal their structure, their architecture to understand their mechanism. And by working with different families of channels and transporters, we wish to elucidate the physical and the chemical principle of membrane transport, this essential process. So prior to 2014, my lab has been mainly using X-ray crystallography to study the structures of the transporters exemplified by glucose transporters. So as shown here, we saw a human GLUT1, GLUT3 in 3 different functional states, which allow us to reveal the working cycle, it's known as the alternating access cycle of cellular uptake of glucose. But there is another very intriguing event that my lab has been working on in the past decade, well, since 2007 actually, almost 14 years. So we know that electrical signaling is essential, really essential for the long distance and rapid response. So I always give the example that if you touch a cup of hot water or a flame, you can immediately withdraw your finger, right? Because this occurs extremely fast, mediated by electrical signals. If this this process is mediated through chemical diffusion, it may take days for you to sense that, and take another equal, 1 or 2 days to control your finger to withdraw. And by that time, probably the finger would become like charcoal. So this just to illustrate how important electrical signaling is. Like you can hear me, I can present. For each heartbeat, for the motion, all these physiological processes rely on the very sophisticated process of initiation, propagation of electric signals. So shown here is a highly simplified diagram of action potential. I will not go into details, but just for those audience who are not familiar with neuroscience, so in our muscles or neurons, the membrane has an electric field across it. Is known as the membrane potential. So just keep in mind, it is negative inside at the resting state. This is due to the asymmetric distribution of ions, mostly of sodium and potassium ions. But this electric field is not static. It fluctuates in response to different stimuli. So when it goes up, we call it depolarization, all right? So when it depolarizes, when this membrane potential depolarizes to certain threshold, the sodium channels, the voltage-gated sodium channels, which I will refer to as NAV channels, activate, which it leads to a rapid influx of sodium ion corresponding to this rapid rising phase of action potential. And immediately upon its activation, it inactivates through a process known as inactivation. Meanwhile, voltage-gated potassium channels activate to allow for the efflux of potassium ions, and eventually that lead to the reset of the membrane potential back to resting state. But this is a highly simplified diagram. I mean, it's subject to very complicated regulation. There are some other variations depending on the tissues. So from my description, you can tell how important the sodium and potassium channels are. So I have been focusing on sodium channels, and because of their physiological significance, the 9 subtypes of sodium channels, NAV 1.1 to NAV 1.9, are mutations or misregulations of these channels are associated with a broad spectrum of these disorders, depending on their specific tissue distribution. I will not go into details for each one. Among this, NAV 1.7, NAV 1.8, and NAV 1.9 are known to be associated with pain disorders. So here's the story. Actually, before the pandemic, David probably know that the opiate crisis has always made the headline. In fact, opiate overdose crisis has been claimed as a national crisis in the United States, and it has claimed about like 130 people every day. And this also represented a huge economic burden, as shown here, like up to 78.5 billion a year. I mean, of course, it's not direct cost, but it's associated with the opioid crisis. But what's it for? It is actually for the relief of chronic pain. Therefore, there is an urgent need for alternative painkillers. And in 2006, there was a report in Nature. So a boy from Pakistan was identified who had no sensation of pain at all. So not only him, but I think several of his cousins also had this insensitivity to pain. And genomic analysis pinpointed the mutation to one gene, SCN9A. And if you remember, this actually encodes the channel NAV 1.7. And that was in 2006. In fact, 2 years before this report, several groups had independently reported the mutations in the same gene, SCN9A, that or associated with extreme pain syndrome. So now you can see like loss of function of this mutation usually is like nonsense mutations or associated with pain insensitivity, and other mutations that lead to probably higher expression or abnormal activation of the same gene can cause like extreme pain disorder. So this evidence, this genetic evidence, has made NAV 1.7 a target for the next generation analgesics or painkillers. And because like the 9 subtypes of sodium channels are so closely related, when you try to develop inhibitors targeting NAV 1.7, you want to make sure its function is specific. It will not suppress your heartbeat, right? That's why we set out to solve the structures of different subtypes of sodium channels in the hope to identify subtype specific druggable sites. And so sodium channels actually belong to a superfamily known as the voltage-gated ion channels that also consists of like calcium channels, potassium channels, and even trip channels, although not all trip channels respond to membrane potential. And so as a structural biologist, I have 2 very fundamental questions to address. So first, like what determines the ion specificity, the ion selectivity, right? So why sodium channels only permeate sodium, but not like the other monovalent like potassium or like calcium, which has a very similar size with like sodium. And a more intriguing question is how do these channels detect the fluctuation of membrane potential and respond accordingly? So prior to structure determination, sequence analysis of the voltage-gated ion channels have already shown that they contain like 4 repeats or 4 subunits. So that made them either homotetramer, homodimer, or just like one polypeptide chain that contains 4 homologous repeats. And within each subunit, there are 6 transmembrane helices known as S1 to S6. And among these 6 segments, the 4 sets of S5 and S6 enclose a central pore that is responsible for the selective ion conduction gating. And the first 4 segments, S1 to S4, each constitute a so-called voltage sensing domain or voltage sensor. And among this, S4 is the most unique one in that it carries repetitively occurring positive charged residues like lysine and arginine. So these basic residues are known to be the gating charge residues. So our knowledge on the structure and mechanism of voltage-gated ion channels actually first came from the study of potassium channels by the pioneering work of Ron McKinnon, the Nobel laureate in 2003. So shown on the left is half is Nobel winning structure actually, but it is only half of the pore domain. And this is the side view. And on the right is the top view or extracellular view of the voltage-gated potassium channel. As you can see, 4 voltage sensors surround a central pore domain. So now, like just to summarize what I've told you, for a voltage-gated ion channel, we know it requires at least 3 elements. You need a selective filter to discriminate different ions. You need the intracellular gate or activation gate to control the opening or closure of the channel. And you need a voltage sensor to detect the membrane potential change. So these are the 3 elements, but in different types of channels, they are different. The details are different. So, and in contrast to the relatively rapid progress in the structural study of the homotetrameric potassium channels, that of the sodium-calcium channels has been slow. Why? Because they are made of a single chain, and they are subject to heavy glycosylation or phosphorylation, which made them heterogeneous. So it is extremely difficult to obtain the recombinant protein for crystallization. Therefore, for a long time before 2017, we had no idea about the structure of eukaryotic single chain sodium or calcium channels. So the shown here was one published in 2000, but in retrospect, this low resolution negative stain EM reconstruction really didn't reveal anything, and it was even misleading. But prior to the study of eukaryotic channels, there were several structures of bacteria sodium channels, but they are made of homotetramers. My lab also contributed. That's our first publication on the sodium channel. But I will not go into details. Just to cut the long story short, the knowledge derived from the bacteria sodium channels, which are homotetramers, or derived from potassium channels, which are also homotetramers, cannot explain, several fundamental questions that are unique to eukaryotic sodium and potassium, and calcium channels. So just to list them, they are made of single chain. So the very fundamental question, how are the 4 repeats organized, remain unclear actually, because theoretically, if they all observe a domain swapped arrangement of the pore domain and voltage sensing domain, there can be at least 6 combinations. And also, among all the voltage-gated ion channels, the sodium channel is most unique in that it contains 4 distinct residues that control ion specificity, DEKA. How these 4 residues collectively select sodium over other ions also remained unclear. And, because this channel is made of single chain, it was already shown through biophysical approaches that they do not activate simultaneously. In fact, the first 3 repeats activate faster than the last one. And activation of the last repeat 4, actually leads to another important process known as the rapid or fast inactivation. So as shown here, this is a typical recording that, once the channel activates, it can inactivate within just 1 millisecond. This is important to ensure, action potential it is like a wave. This is important to ensure the repetitive occurring of the firing. So how is the channel achieve fast inactivation? Ever since 1970s, it was already shown by Armstrong and Bazania. So it's Clay Armstrong and Pancho Bazania. My pronunciation may not be quite right. That the intracellular segment controls the inactivation. And then later on, Bill Catterall showed that a small cluster containing 3 hydrophobic residues, IFM, as leucine, phenylalanine, and methionine, shown here actually as this ball, executes fast inactivation. It is on the linker between repeat 3 and repeat 4. So this model was referred to as ball and chain model. It was quite neat. Unfortunately, this model actually contradicts with a number of experimental evidence. So exactly how sodium channels achieve fast inactivation also remained to be elucidated. And this slide may be interesting to David, right? Because, you study a lot of toxins, and sodium channels are actually the primary targets for many natural toxins from the animal venom. And in fact, before the structure was available, 7 sites for different types of toxins, either small molecules or peptide toxins, have been identified and mapped to the primary sequence. Unfortunately, these sites cannot be recapitulated on the model of potassium and bacteria sodium channels. Therefore, a structure of eukaryotic sodium channel is really required to understand the action of these toxins, and also local anesthetics, some FDA-approved drugs. So, okay, this is the end of my introduction, 20 minutes. For a long time, we have been struggling with the structure, expression, and production. So I will just, cut a very long story short. So since 2012, I'm really grateful that I had the privilege to work with the young scholars listed here, not all of them, but the major contributors to the study in Tsinghua. We were able to solve the structures of a number of sodium channels. So, but prior to that, because of the fantastic cryo-EM facility that was initiated in 2009, way before the revolution of cryo-EM, we were able to solve the structures of a few much larger channels like the radioreceptors and the multi-subunit calcium channels. So during this process, we also trained ourselves to get familiar with cryo-EM. And then, we began to attack our top target, the sodium channels. So they are much smaller. They have a pseudo 4-fold symmetry. Technically, they are more challenging than the rounding receptor, which are gigantic homotetramers, or the CAV 1.1. So these are a few structures that we've determined in the past since 2017, but the struggle started in 2012, in terms of eukaryotic sodium channels. So just, I know in the audience, quite some of you are students. So I'd like to share some technical aspects, although it will be pretty fast. So the first, as I said, bottleneck for structure determination was to obtain, the proteins that are suitable for cryo-EM imaging. And for a long time, we've been struggling with human proteins. And then a very talented graduate student, Huaizong, who is now a principal investigator in Westlake University, came up with an idea. It was like, it's so difficult to get these proteins. Maybe it's because of the intracellular domains. Why don't I just look for some short isoform among all the eukaryotic sodium channels? And, with this, he actually just searched all the data bank for all the deposited sequences of sodium channels and identified one from American cockroaches. So we named this one NAVPAS. S stands for short. And indeed, now we can see a decent peak on SuperDAX. And also the peak looks decent, right? But if you really calculate the yield, it was very low at, in our opinion at that time, like only 50 micrograms out of 8 liters. But now, after we've been working with human proteins, this was really a high yield channel. But this was only the first step. After we got the protein, fortunately, this protein can easily get into the vitreous eyes and, distributed with different orientations. So we did not have to struggle with like cross-sample preparation. But then because of, its small size and because of the pseudo four-fold symmetry, we've encountered a second barrier, like the data processing cryo-EM. It really took Qiang Zhou, who was trained by Professor Senfang Sui and was a research track associate professor in my lab at that time. And it took him several months just to figure out a way, a protocol, a new procedure to process the data. So in retrospect, his struggle really gave us a fantastic workflow. So later on, whenever we started data processing with sodium channels, we follow the same protocol, and it worked like a charm. To cut the long story short, we were able to solve the structure of, the first structure of eukaryotic sodium channel. And as shown here, it contains like 4 repeats. And, the major part is transmembrane region. And on top, it is extracellular segments. You can see, the several glycosyl moieties were revealed. And on the intracellular side, there are also several domains that I will come back later. And then, but unfortunately, for this channel, which was from like American cockroach, it cannot be recorded. We could not obtain electrophysiological data. So this made it, hard to establish the structure and function relationship. Therefore, we switched to human protein again. So this time, we thought after the structure determination, the first one should be easy. It turns out it's not. So another graduate student and then a postdoc, Xiaojing Pan in my lab, really spent several years. She started this like in 2012. It took her, 5 years just to struggle for protein expression. She tried different systems, different constructs, different detergents, and eventually it is actually the so-called BAC-MAM system that you infect mammalian cells using baculovirus that gave us, a decent yield, like 40 to 60 micrograms per 12 liters. Also, this now, in retrospect, was still a high yield protein. All right, but after we got the protein, so this is the skeletal muscle isoform is known as NAV 1.4, but it is so difficult to get this protein into the vitreous eyes. As you can see, they tend to either aggregate in thick eyes or like you see no particle in thin eyes. Xiaojing really struggled for a long, long time. And one day I was just like, discussing with her, I was like, hey, come on, we were trained as crystallographer, right? We're not strangers to screening methods. Why don't you get a crystallization kit, which was shelved for a long time, and see, what conditions you can add as additive? So, Xiaojing really took my advice seriously. So she screened, she made a matrix to screen like salt concentration, different salt, and different pH. She started with pH, and to my surprise, actually this was an important factor. So this shown here are SuperDAX like chromatograms, and you can see at different pH, the width of the half peak is different. Although, for most people, this may be nothing, but for well-trained biochemists, this means a lot. That really reflects the sample quality, whether they are homogeneous enough. And also, so Xiaojing just selected pH 6.0 to be the optimal one. And even at this specific pH, she tried different buffers, but different chemicals as buffering regions. And turns out imidazole improved the particle distribution. And then after we identified this condition, we searched literature. Turns out there was already report that imidazole could also help improve the cross-sample property in another case as shown here. But then we still had some problem with the probably air-water interface. So Xiaojing and other junior students also screened for different, like, even cross-linkers or like different types of grays, and also some surfactants. Eventually, addition of extremely low concentration of SDS helped to further improve the cross-sample quality. So eventually, Xiaojing obtained very decent electron micrograph, and solved the structure of the first structure of human sodium channel, which has been very well characterized. Therefore, we now have a model for structure and function relationship study. So, after this, kind of endeavor, I coined a term, proteinality. So that means every channel has its own specific, unique proteinality, just as like every person has your own personality. All right, but so after that, we solved several other human channels, sodium channels. To our probably disappointment, so far among all these channels, we only captured the two major conformations. One represented by the first nav path structure, and all others exhibit very similar conformations. The following discussion will be based on this two conformations. So what have we learned from these structures? Remember, the first question, about selective filter. Sodium channels contain 4 distinct amino acids, D, E, K, A, to determine the sodium selectivity. So prior to structure determination, we were thinking probably this lysine would alternatively form, interact with D or E, to achieve some sort of alternating access in the select filter vestibule. However, to our surprise, after we solve all the structures, this lysine is extremely stable. That means it does not move at all. In fact, when we collaborate with Haipeng, a professor, now like a professor in Tsinghua, and his student, Juanrong Zhang, we identified a potential permeation path for sodium through the selective filter. So we also identified a relatively stable sodium coordinating site constituted by DE and another invariant E in the second repeat as shown here. So this molecular dynamic simulation performed by Juanrong showed this permeation path, and just a few months after this discovery, we actually solved the high resolution structure of sodium channel at 2.6 angstrom, and this time we could see a density that very much likely belongs to a sodium. As you can see, indeed, it is it is just consistent with the computation. So this DEE site provides a relatively stable coordination of sodium, and during the permeation, the lysine and alanine in repeats 3 and 4 just stay stable. It seems like their role is just to repel this cation to the other side. So that's our current understanding of the selective filter. And in terms of intracellular gating, because we only obtained 2 conformations, unfortunately, the diameter of their intracellular gate varies a lot. So at the beginning, we thought we've already captured a kind of activated channel with a radius of about 2.7 angstrom. But then also through MD simulation, we realized that this is not wide enough for a hydrated sodium ion. So far we haven't captured an open state. Nevertheless, the conformational change between these channels gave us a sense of how the pore domain may undergo conformational changes to open this gate. As you can see, this helical bundle undergo an iris-like rotation. Each helix S6 also undergoes axial rotation. And during this process, shown here is a surface view of the pore domain, side view. You can see actually on the side, the wall also seems like it's gated, right? So if it opens, it's called a fenestration. And, in that path, there's no fenestration at all. Whereas in other channels, we can see these big fenestrations. And also this site, this particular one, enclosed by the repeats 3 and 4, was already known to be, the binding site for local aesthetics. So therefore, this shows that, the pore domain also undergo conformational changes. This may represent potential druggable site for subtype-specific inhibitors. All right, so the next part is my most favorite part about the fast inactivation. So just let me remind you again. So this channel undergoes, quick inactivation upon its activation. And so the previous model was like a ball and a chain model. So this model was originally proposed for potassium channel and also borrowed by sodium channel. But until last year, there was no structure of a ball and chain inactivation of potassium channel either. And last year, Karina Nimijan's group from Will Cornell actually solved the structure. And you can see this is the so-called ball. It inserts into the pore domain from the intracellular gate, right? But after we solve our structures of sodium previously, the calcium channel, to our surprise, this 3,4 linker, which carries the fast inactivation motif, actually in both structures, it is sequestered by a globular C-terminal domain. And the IFI motif is placed really far away from the intracellular gate. So how to reconcile this? So we were like, the previous model may be wrong. Fortunately, we did put that in our paper. So after we solved the structure of like NAV 1.4 from first electric eel and then human, we observed a pronounced conformational change of this 3,4 linker. So this can be better illustrated in the morph. As you can see, this is the original position in NAV path, and the other structures, this 3,4 linker undergoes marked translocation to place the IFI motif into the pore domain. But it is not like the potassium channel where the intracellular gate is blocked. Rather, this F motif is inserted into a cavity that's outside of this intracellular gate. It's outside of the tetrahelical bundle that encloses the gate. So this reminds me of the door wedge, right? If you think about it, if you want to close a door, there can be two ways. One is you directly stand there to block it, and another is you push the door to close. And by squeezing the door to close, we have this, this channel has the advantage of avoiding leakage, right? Because, if you just block this intracellular gate, when you remove the blocker, there can be potential ion leak. So that's why we propose this allosteric blocking or door wedge model to account for fast inactivation. And, this model that was first observed in the structure of electric eel sodium channel, we proposed in 2017, was recapitulated in all human channels, also like last year by Bill Cattrall's group. And also it is consistent with, tremendous amount of mutagenic study. And recently we systematically analyzed some disease mutations, and indeed dozens of mutations that are associated with the lung QT syndrome in NAV 1.5, the cardiac sodium channel, are either directly or indirectly involved in the segments for fast inactivation according to our door wedge model. Okay, so how long do I have? I have 8 minutes. So the next part, if you remember, I told you that sodium channels are the primary targets for toxins. So for this part, I will go faster, probably. There are two prototypical small molecule inhibitors, the tetrodotoxin and saxitoxin. So they are actually the two molecules to characterize sodium channels. So just to cut the long story short, we were really extremely happy that DM is so powerful that it allowed for the resolution of small molecules like TTX or STX. These structures clearly illustrated why some sodium channels can be resistant to these small toxins. like this movie. And another type of toxin is peptide toxin. So here is a clip from the video from like a Chinese male bat swallow fish. So you can see it looks like a needle, right? To stab the fish. Actually, it is like, it injects venom into the fish to paralyze it. And the main like one of the main constituents in the venom is peptide toxin known as the mucotoxin K3A, which I will refer to K3A, which is like 16 amino acid peptides that contains 3 pairs of disulfide bonds. But for a long time, how this toxin blocks sodium channel remains unclear. So, well, we solved the structure of K3A bound to NAV 1.2, a central nervous system sodium channel, and as you can see here, so this toxin sits right above the selective filter. So when we compare this one to a previous structure of potassium channel in complex with another toxin, although toxins are different, they have different length or composition, but we identified like one common paradigm. So both toxins contains one lysine that just projects into the mouth to the selective filter. So this clearly immediately reveals how the toxin blocks ion conductance. And another type of peptide toxin is known as the gating modifier. I will not go into details. Basically, we solve the structures of potassium channels in complex with different types of GMT, gating modifier toxins. I will not go into details, but just to tell you what we've learned. So for these toxins, for the gating modifier, so they require, part of this toxin requires an anchorage, either like a stable pore domain elements or the membrane. And then the other part interferes with the voltage sensor to control the movement of the voltage sensor. So that's what we've learned from the structures. Okay, I hope to spend the next 5 minutes. I will skip the summary part, like, what we've learned about selective filter, or like, map the disease, mutations, like how, the structures in different conformations may facilitate mechanistic understanding and drug discovery. But just let me tell you, like, our current challenge. So as I said, like for the so the process of voltage sensing to the gate opening or closure is known as electromechanical coupling. So to understand this process, we still need two structures for the minimal. One is the channel at the resting state. Another is the one in the activated states. But for resting state, we've tried. You cannot imagine how many mutations or like chemical modifiers we've tried. But the structures always remain the same, like the inactivated states. So we were like, why don't we invent some new method just to apply electric field to the channel directly. So when I moved to Princeton in 2017, this has been my major research aim, like to invent like a new method for cryo-EM study, like to create more conditions for sample preparation. So to introduce a membrane potential or transmembrane electric field, there can be two potential strategies. One is you embed this protein into a liposome and you try to manipulate the ion distribution, just as in the cell, right? Cell use like ATP pump, sodium potassium pump, to maintain the asymmetric distribution of sodium potassium ions. So that's one strategy. Another strategy is you directly apply a physical electric field, through a device to your sample during this plunged freezing. So we actually followed both strategies, and we are still in the middle of this endeavor. So for liposome embedded protein, there are several, again, technical bottlenecks. So first is like, how can you, reconstitute structure out of proteins embedded in liposome? So Xia Yao, who got her PhD with Professor Yu Rao in School of Pharmaceutical Sciences in Tsinghua. Actually has been focusing on this. I will not go into details. Basically, she figure out she refined the protocol, and she were able to obtain beautifully distributed liposome on the cryo grade. But during this process, we realized that we need graphene grades, and then for even distribution of the liposomes. But for graphene grades, so for a long time, we could not obtain high quality graphene grades from commercial resource. So actually, when my first postdoc I recruited to Princeton was Dr. Yimo Han. She actually got her bachelor's degree from 数理基科班 in Tsinghua University. She is now an assistant professor in Rice University. So she got her PhD in nanomaterial science from Cornell University. And when she joined my lab, we have been focusing on, on the generation of high quality and easy to use graphene grades. So we published, we deposit like the video to YouTube, and also, in the PNet's website, you can find a detailed protocol of how to make this high quality, high yield, and cheap graphene grades. So basically with this, we were able to, obtain high quality cryo sample, of like membrane proteins embedded in grades. And then Xiao Fan, who actually spent 9 years, or even 9 years in Tsinghua, he was trained by Professor Hongwei Wang for a PhD study, and work out the workflow for data collection of membrane proteins embedded in liposome. So we used ACRB as a prototypical protein as a 2 molecule. So the reported structure in that paper was like 3.8 angstrom, but now it can reach like 3.0 angstrom. So with this, the advantage is we could even observe like the curvature of the membrane. So we think this method, although we haven't applied an electric field, this may help the study of membrane proteins that interacts with the membrane for like either mechanical, through mechanical force, or like its conformation is impacted by the transmembrane ion gradient. So we are now in the process of introducing an ion gradient to the system. So all right, so maybe for long run, our goal is not maybe. So our long-term goal is to reveal like the structures of high resolution structures of sodium channels in vivo to to elucidate its distribution in the neurons, and also to understand its crosstalk with other, among themselves and with other proteins or channels. So that's my long-term goal. With that, so as you can tell, really, my heroes for these achievements are all directly or indirectly related to Tsinghua. It's really a Tsinghua team, either my students in Tsinghua or like all the alumni of Tsinghua. I think, this is probably a good time. I hope like, this presentation is some sort of celebration to Tsinghua for its 110th anniversary, and also to our department, or now School of Life Sciences, for its 95th anniversary, and also for my own class. Like I was enrolled in 1996. It's like the 25th anniversary of birthday. So shown here, when I was preparing for this slide, I found this old picture. It is of like the group from my class, and also the class just 1 year ahead of us, by 95. And interestingly, I found the 5 structural biologists. So see, the audience, whether you can recognize them. Our host, Hongwei Wang, who was my actually mentor, the mentor to our class. And also, I'm not sure whether David will mention this guy, Maofu Liao, who is now associate professor in Harvard University, and Liang Feng, who is tenured associate professor in Stanford University. And probably like HYPES represent like kind of top university in the United States for undergrads. This is some one that you may not be able to recognize. And this one, Yanhui Xu, who just recently published a beautiful structure, gigantic complex regarding like initiation transcription. He's now in Fudan University. Then I realized, among this huge group, only this small area, like we were concentrated in this small area. So this kind of polarization of memory analogy. So if, in the audience, the students are postdocs, if you want to become a professor in structural biology, go to Donglingshan and find this spot to take a picture. Maybe in 20 years, you will give a similar talk, not similar, of course, fancier talk in structural biology. So with that, thank you so much. Thanks again for this great opportunity. Take this opportunity as part of the celebration to Tsinghua and our department. So I'm ready to take questions.
Thank you, Ning. That's a wonderful talk, and especially a lot of memories, which is very important based on the channels. Yeah. So we can take some questions. I see there's already a question from the chat room. First one, I guess it's about when you're talking about the reaction of animal, asking about tree sloth.
Lightning.
They move really slow no matter what happens. So what happened to their neural system?
So we see a fan of like a Zootopia, like the sloths. Yes, but I think it's under control whether they move faster or slower, right? Although you think they move slow, but keep in mind their hearts still beating, right? In terms of electric signaling, it's still fast, regardless how slow their motion is. And the second question, is there any rationale about screening condition for cryo-EM sample prep? I think so. As I said, when we realize, the protein yield is low, we attribute it to folding. And in order to make the folding easier for our target, we set out to look for shorter subtype that may contain, less flexible segments. So this worked. I think this actually follows rationale. And for water-air interface, because this is attributed to water-air interface, and at that time we were not quite familiar with graphene grids yet. Otherwise, we would have tried graphene grids, but we did not have high-quality graphene grids. But because it was kind of air-water interface, so we tried like the surfactant, some detergents, so that also helped. And Junjie, hey, congratulations, Junjie, for your new chapter in your career.
The major question is following two, about the evolution of the nerve channel.
So the first question is, any evolution knowledge about how the sodium channel get the 4 repeats? Is there a crosstalk similarity between sodium and other channels on the gene side, or share the same ancestor? That's a good question, because we know bacteria sodium channels are homo tetramers, and potassium channels are also homo tetramers. So there was quite some debates about which came earlier, sodium or potassium. But for sure at the beginning, or not for sure, because we have we know there are bacteria homotetramers, so probably sodium channels also started with the homotetramer, and then because it requires 4 distinct residues for sodium selectivity, probably at the beginning you can imagine subunits with different residue at the select filter assembled together. But then during selection, I think it's because of this asymmetry that's required for sodium selection that eventually a single chain has been evolved. So that's my naive interpretation about evolution. And about crosstalk, yes, for sure there are crosstalk, but compared to calcium channels, our understanding of sodium channel within the membrane is still limited. It was shown that, it was suggested that the sodium channel may be able to dimerize, homodimerize, but there was no solid evidence. And they certainly interact with a lot of proteins and tethering factors, so that remains to be elucidated. The second question is why life chooses to mainly use cation but not anion channel for cell membrane signaling. I guess it's because of the abundance of ions. Very interesting. The other day, Chen Song from Peking University discussed with me about why there was no specific lithium channel, and I was like, maybe it's because of at the beginning, on Earth, life. We don't know, but probably life came from seawater, right? If you not calculate, but at least now, from our current understanding of the ion concentration in ocean, like sodium is most abundant, followed by potassium and calcium. And of course, there are also chloride, maybe because like chloride is dominating. There's this one specific one, and also because you study RNA and think about the charge DNA and RNA carries, right? So chloride may be required for, the electric shielding. So I think those are interesting questions, but I believe in selection during natural selection during evolution. So I think mostly it's because of the ion concentration abundance, as well as the impact of the kind of the charge within the cell. Take DNA and RNA into consideration. Next question, how to improve the map quality of the gate modifying toxin bound to sodium channel to review more detailed binding sites? That's a very good question. So our current, as I said, my group's understanding of this is that it requires, so far we've solved the structures of sodium channel in complex with, I would say, 3 types of toxins. Like for Pol blockers, they are very well resolved. And for the modifier, getting modifier toxins, like as long as you can identify its rigid scaffold to stabilize the toxin, it is well resolved. But for those that require membrane, because like during protein purification, the membrane is disrupted, so the toxin became flexible. So we think the way to go, one from our attempt, we tried nanodisk, but for eukaryotic sodium channels, the protein yield is a limiting factor. For the current yield, there's no way to reconstitute the sodium channels into nanodisk. So that's why, like another Tsinghua student, Shuai Gao, who got his PhD from Liu Lei, from chemistry, actually engineered a chimeric protein with a bacteria natchback. So bacteria sodium channel as the scaffold, and grafted the toxin binding site from human sodium channels to interact with the toxins. So for that chimeric protein, we were able to incorporate that into nanodisk, and indeed the resolution was improved. But I think it really depends on the state of the channel. If the toxin prefers inactive state, maybe there's a good chance to capture a high resolution toxin. But if it prefers kind of a resting state, I guess the only solution is to get the right state, and then the binding can be highly specific, and then the toxin can be better resolved. Yeah, that's something we are still working on.
Actually, I have a question regarding the last part of your talk. By reconstitute the channels into liposome, and you showed a nice 2D cartographies of the particle embedding in the vesicle, and seems that the particle the orientation is inside the vesicle. I'm wondering if you see some particle also with the orientation sitting the opposite way.
In fact, at the beginning, we thought, this protein has preferred orientation. Turns out it has preferred selection of the particles. Then, in the past year, we've been collaborating with Zhang Xianzheng from IBP, using his algorithm, to reprocess this data. We actually obtained class of higher resolution for the channel with the other orientation.
And then the problem is, if you want to apply the for instance, the ion gradient onto this kind of episode. If you have a different orientation of the particle, would that be a problem for you?
That's actually, I think this is an advantage, right? That you can capture like the channels, because classification is powerful. You can discriminate these two classes, right? Some some channels are in the kind of positive potential, some in negative potential, and luckily, if we are lucky enough, we may be able to capture two distinct conformations. So I'm not worried about it.
Mm. Do we have more questions? Okay, if no